Okay, so after respiratory system, in histology, the next topic which you have to study, that is the GIT. Okay, so in GIT, if you see the entire GIT, there are 4 basic layers present in the GIT. It is common. Okay, for example, if you see the structure of esophagus, if you see the structure of stomach, small intestine, large intestine, these 4 layers are present. Okay, but there will be some differences. As you will go downwards, you can see the differences, but these 4 basic layers are present everywhere. Okay, so that is a speciality of the GIT. Okay, now if you see the layers, so in GIT, first today we'll discuss about the basic layers of the GIT, or you can say basic structure of the GIT. Okay, what are present in all those layers, and then what are the different types of modification present in these layers. Followed by, we'll discuss in detail about the esophagus and stomach. Now in general structure of GIT, as I said, 4 layers are there. What are these 4 layers? Mucosa, submucosa, muscularis, externa, adventitia or serosa. Okay, these are the 4 layers. Now if you see the mucosa, again 3 additional layers are there, or you can say these 3 layers together, they are forming the mucosa. Okay, what are they? First is the lining epithelium, lamina propria and muscularis mucosae. These are the 3 layers together, they will be forming the mucosa. Okay, next, submucosa. Now mostly in submucosa, dense irregular connective tissue is there, and along with that a nerve plexus is present, and that is called as Meissner's plexus. Anyone, have you heard about this Meissner's plexus? Someone is saying yes, someone is saying no. Yes, I know you have heard about Meissner's plexus. What is the function? Two types of plexuses are there, or you can say 2 plexuses are present in the layers of the GIT. In submucosa, the plexus which is present, nerve plexus, that is called as Meissner's plexus. And in case of muscularis externa, another plexus is there, that is called as myenteric plexus, or also called as Ayursvech plexus. Function kya hai? What is the function? Very simple. In case of submucosa, the nerve plexus which is present, it controls the secretion. It controls the secretion. Along with that, it also controls the absorption. And the nerve plexus which is present in the muscularis externa, okay, it controls the motility. We know that peristalsis movement is taking place, that is controlled by the nerve plexus which is present over here. Okay. Now this nerve plexus which are formed, 2 components, both sympathetic, parasympathetic are there. So sympathetic nerves will be there, mostly postganglionic, and in case of parasympathetic, the parasympathetic ganglia are present. Okay, so the fibers which are coming mainly from vagus, they will rely on the ganglion which are present in this particular plexuses. Okay, so that is the preganglionic part, and from there fibers which are coming out, that will be the postganglionic part. And this entire part comes under enteric nervous system, ENS, or also called as enteric nervous system. Okay, details we'll discuss in head and neck portion, don't worry. Okay, so in submucosa, dense irregular connective tissue and Meissner's plexus is there. Next, muscularis externa, very important layer, because this particular layer is controlling the peristalsis movement. Okay? Now here we are having smooth muscle. Okay, but entire part smooth muscles are not there, some exceptions are there, that also we'll discuss. Now smooth muscles are mainly present. Now how the smooth muscles are arranged? Okay, single layer of smooth muscle are not there, multiple layers of smooth muscles are there. Most of the cases 2 layers of smooth muscles are there, as you can see, that is inner circular and outer longitudinal. Okay, inner circular and outer longitudinal, but there are some cases where an additional layer of muscles are also present. Lastly, the adventitia or serosa. Most of the cases it will be serosa, and in few cases it will be adventitia. Okay, where connective tissue is there, if it is adventitia, and if it is serosa, it is covered by peritoneum, that is nothing but the simple squamous epithelium, or also called as, anyone, mesothelium, also called as mesothelium. Now what is the difference between the adventitia serosa? slash which is given doesn't mean that adventitia and serosa are same नहीं है. What is the difference? Difference is the part which is covered by peritoneum, part of the GIT which is covered by peritoneum, that is called as serosa. The part which is not covered by peritoneum, that is called as adventitia. Can you give one or two examples? Yes. Bare areas are also there. Okay. So in case of bare area, you will be getting the adventitia, and where the peritoneal folds, mesentery or other things are attached, this will be the serosa. Okay, so these are the layers. Next, this is a 3D diagram. This you don't have to draw. This is 3D diagram for your understanding. Okay. Now can you see all the layers one by one? ये देखो. This is the mucosa. So in mucosa is made up of how many layers? 3 layers are there. First is the lining epithelium, as you can see. Next is the lamina propria, followed by the muscularis mucosae. Now in muscularis mucosae also smooth muscle cells are present. Okay. Now your question will be, in muscularis externa, 2 layers or sometimes 3 layers of smooth muscles are there. Then why smooth muscles are also present in muscularis mucosae? Any idea? See, the smooth muscles which are present in the muscularis mucosae, what is their function? Their function is it will help in mixing the food with the secretions which are coming out from the different parts of the GIT. Okay, we know that in different parts of the GIT cells are there from which secretions are taking place. For example, gastric glands. Okay, gastric secretions are there. If you go to the duodenum, secretions are there. Okay, so that secretion, why that secretion is important? It has to mix with the food, then only the proper digestion will take place. Now whenever you are eating something, you used to mix with your hand. अंदर जब जाता है कैसे हाथ डाल के मिक्स करोगे? Not possible. तो कैसे मिक्स होगा? फॉर दैट दिस मस्कुलरिस म्यूकोजल लेयर विच इज देयर, उसका जो एक्टिव कंट्रैक्शन रिलैक्सेशन होगा, दैट विल हेल्प इन मिक्सिंग ऑफ द content which is secreted and the food, so that the digestion can be proper. Okay, everyone, have you noted down the layers, name of the layers? Okay, now here you can see these 3 layers. First, lining epithelium is there, lamina propria, muscularis mucosa, and as I said, in muscularis mucosa also smooth muscle fibers are present. Okay, next if you go below that, then you can see the submucosal layer. See, this is the submucosal layer. Now there are some areas of the GIT where different types of glands are present in the submucosa. Okay, for example, in esophagus, esophageal glands are present. In case of duodenal glands are present, or also called as Brunner's gland. Okay, in submucosa, these are the modifications of some part of the mucosa, submucosa. Next, after submucosa, as you can see, muscularis externa is there. Now in case of muscularis externa, mainly 2 layers of muscle fibers are there, that is the inner circular and outer longitudinal, which you can see. Okay, additionally there are some cases where another layer is present in between these 2 layers. Okay, next there are some areas where modification of this particular circular layer takes place. For example, pyloric sphincter. I hope you have heard about pyloric sphincter. ठीक है, तो जो pyloric sphincter फॉर्म होता है, कैसे फॉर्म होता है? It is because of the condensation of the circular layer in that particular area. Okay, the sphincters are formed. Okay, so like this, some areas are there where these modifications of the smooth muscle layer takes place. Followed by the serosa or adventitia. Already we have discussed about the difference between serosa and adventitia. Okay, serosa means it will be covered by peritoneum. Adventitia means connective tissue where peritoneum is absent. So now we will discuss about one by one. And next, in order to increase the rate of absorption, there are some modification. Already I have mentioned few modification. First modification is the surface area and the length. So you have seen that the length of the GIT, okay, is extensive, so that the absorption can take place properly. Next, the presence of numerous folds involving the entire thickness of the mucous membrane, that we have discussed in case of internal structure of duodenum. Do you remember? In the internal structure of duodenum, I told you circular folds are there. What is the name of the circular folds? These are called as valve of Kerckring, or also called as plica circularis. They also play important role because due to the presence of that circular folds which are there, surface area increases. As a result, the food which is passing through it, it will get delayed, so that it will get enough time to do the digestion and absorption. Now these folds can be seen easily. Okay, so whenever you are dissecting the intestine, please see the folds. It can be seen easily with the help of normal eye. Next, numerous places epithelium dips into the lamina propria. Okay, so what are the modifications present in case of lamina propria? Lining epithelium will dip into the lamina propria. Okay, and that is called as crypts. Okay, these are called as crypts. In case of stomach, these are called as pits. Next, in small intestine, mucosa bears finger-like processes, that is called as villi. Okay, so structure of villi you have to study in case of intestine. Finger-like processes are there in the mainly in the intestine, and these are called as villi. Next, each villus is having surface lining of epithelium, and core is having extension of the connective tissue from the lamina propria. Okay, which helps in the absorption. Next, the luminal surface of the epithelial cell bears numerous microvilli that also helps in the absorption. Okay, so these are some modifications which you can see in the mucosa of the various part of GIT. Now, epithelial also perform very important secretory functions. Secretory cells are arranged in different types of glands. Okay, so in stomach also 3 different types of glands are there. Next, in duodenum also duodenal glands are present. Okay. The lamina propria, as you know, it is a loose connective tissue. If loose connective tissue is there, that means we will be getting the blood vessels, lymphatics, etc. Okay. Now the lymphatics which are present, especially in case of small intestine, okay, the villi which is present, in that centrally placed lymphatic channel is there, that is called lacteal, which helps in the absorption, mainly the fat part of the meal which we are having. Whenever we are eating something, at that time protein is there, carbohydrates are there, fat are there, right? So जब absorption होता है, the fat part which is absorbed, that is absorbed by the lymphatics. बाकी जितना भी absorption होता है, that is absorbed by the capillaries which are present in the lacteal, in the villus. But only the fat is absorbed by the lacteal, that is the central lymphatics. Okay, that is the difference. Next, the lamina propria is associated with GALT. Now, what is this? Do you remember GALT, which we have studied in trachea, bronchus associated lymphoid tissue? Similar aggregation of lymphatic follicles, lymphatic tissue are present in case of in the lamina propria of GIT, and that is called as GALT. That is gut associated lymphoid tissue. Okay. So, what will be the role? Major role is protection, phagocytosis, antibody secretion, etc. Next, in case of ileum, the lymphatics nodule occupy the lamina propria in a larger extent, and that is called as Peyer's patches. Okay, that also can be seen easily under compound microscope, Peyer's patches. Okay. Now, the lamina propria also provide mucosal depression, which form the glands. In case of stomach, it is called as gastric glands. In case of intestine, it is called as crypts of Lieberkühn. Okay, these are some speciality of the lamina propria. Next, muscularis mucosa. So, muscularis mucosa is a thin layer of smooth muscle. Okay, smooth muscles are there, but very thin layer. What is the main function? It separates the connective tissue of the lamina propria from the submucosa. Okay, so submucosal layer, lamina propria from the submucosa. Okay, so the lamina propria which is there in the submucosa, that is separated from each other by this muscularis mucosa, and this muscularis mucosa is a thin sheet, or you can say thin layer of smooth muscle fibers. Now, already I have told you the functions. Now, it consists of inner layer, okay, which are arranged circularly, and outer layer which are arranged longitudinally. So here also the arrangement is there, inner circular and outer longitudinal, and this particular layer is well developed in case of stomach. Now, the muscularis mucosa extend into the mucosal folds, but not into the villi. That is one important point, and this contraction of this particular layer helps in local mixing of the content. Okay, so whatever the content, whatever secretions are there, okay, the mixing with the food is taking place with the help of this particular layer. muscularis mucosa. Next is the submucosa. So submucosa is made up of dense irregular connective tissue. Now dense connective tissue is there, that means we will be having blood vessels, lymphatics, and some glands are there. Now these glands are not present everywhere, okay, in some areas only these glands are present, okay. Now where exactly these glands are present? It is present in esophagus and duodenum. Today we will see the esophageal glands, okay, and in the next week we will see the glands which are present in the duodenum. Next, this is very important, submucosa also shows a presence of unmyelinated nerve fibers and ganglion cells to form submucous Meissner's plexus. Okay, plexus of nerve, where we will be getting the ganglionic cells or ganglion are present, and along with that unmyelinated nerve fibers are also present. Okay, now what is the function of this Meissner's plexus? It controls the secretion and absorption. It controls the secretion and absorption. This submucosa also allows the movement of mucosa over the muscularis externa. Okay, next muscularis externa. So muscularis externa, again it is a smooth muscle layer. Okay, mainly how it is arranged, two layers are there, inner circular and outer longitudinal layer, but there are some modification. Okay, that modification, first of all, if you see the stomach, an additional layer is present in between these two layers. Okay, next if you see the esophagus, okay, if you see the esophagus, the entire, can anyone tell me what is the length of esophagus? Yes, bolo. Approximately. Yes, you are right. 25 centimeter. The length of the esophagus is approximately 25 centimeter. Okay. So if you see the upper part of the esophagus, okay, it is made up of skeletal muscle. Upper part of the esophagus made up of skeletal muscle. Middle portion of the esophagus having combined both skeletal muscle also, smooth muscle also. And if you go to the terminal part of the esophagus, lower end of the esophagus, where it is joining the cardiac end of the stomach, that portion is completely made up of smooth muscle. So the transition is taking place. Okay, that is important. So everywhere in muscularis externa, we are not getting the smooth muscle. Okay, that is the exception in case of esophagus only. Rest everywhere you will be getting the smooth muscle. Okay. Now in case of stomach, inner oblique is there, then middle circular and outermost longitudinal layer. As I said, in case of stomach, not two, three layers are there. First will be the oblique layer, then circular layer, and then outermost longitudinal layer in the muscularis externa. And whenever you are writing it in the exam, you have to mention that stomach is having three additional layers. Okay. Means one layer is extra, that's why three layers are present in the muscularis externa. Next, another important modification is there. What is that modification? In case of large intestine, the longitudinal fibers are prominent bundles and they form a structure that is called as tinea coli. Okay, in large intestine, there is a modification of the longitudinal bundles of the smooth muscle fiber and it is called as tinea coli. Okay. Next, there are some parts where the circular muscle layer, okay, it becomes thickened to form sphincters. For example, pyloric sphincter, ileocecal valve, internal anal sphincter, etc. Okay, that means there are some areas where the smooth muscle layer, which smooth muscle layer, circular smooth muscle layer get thickened to form sphincters. Okay, the best example is the pyloric sphincter. Okay. Next, between the inner circular and outer longitudinal layer, a plexus is there, nerve plexus, that is called as Eyer's plexus or also called as myenteric plexus. Previously, it was Meissner's plexus. Now, this particular nerve plexus is called as myenteric plexus. Iska function kya hai? What is the function? Motility. It controls the motility, mainly the peristalsis movement. Okay. तो यहाँ से शॉर्ट क्वेश्चन MCQ आता है, प्लीज बी केयरफुल, नोट इट डाउन. Now function, as you can see, contraction of the muscularis externa propel the content of the intestinal lumen, okay, and it also controls the peristalsis movement. Lastly, the serosa or adventitia, it is a outer layer of connective tissue the intestine is covered by layer of peritoneum. Okay, this is called as serosa, and there are some areas of the intestine where this serosa is absent, only connective tissue layer is there, that is called as adventitia. Okay, तो adventitia में mainly क्या रहता है, large blood vessels, lymphatics, nerves, etc. These are present. Okay. So these are the layers of the GIT. Okay, so in layers of the GIT we have seen four basic layers are there. In each and every layer, what is present, that also we have discussed, and we have discussed there the modification, different types of modification which are present in the various part of the GIT. Okay. Okay, next we'll start with the esophagus. So you know that esophagus is also the part of GIT. Okay, so though we are in GIT, so we have to study about the GIT also. So if esophagus is the part of the GIT, then how many layers will be there? 4 basic layers will be there. Okay, so any time if the question comes, those 4 basic layers will be present. Please keep that in mind. Okay, there will be some modification, but ये 4 basic layers हमेशा रहेगा. So you can see mucosa is there, submucosa, muscle coat, adventitia or serosa. Now in case of esophagus, I don't know whether you have seen the esophagus or not. I hope you have seen the esophagus. Esophagus तो सबने देखा है. If you have seen the stomach means you have seen the esophagus also. ठीक है. अगर नहीं देखा है तो कल जाके अच्छा से देखो esophagus. When you have studied the mediastinum, at that time have you seen the esophagus? You have not, mediastinum you have studied, you have not seen esophagus. I know you have seen. अच्छा से देखो. Why I am asking? Because of this one. What do you think? What will be present? This one or this one? Huh? What will present in the esophagus? Adventitia or serosa? Most of the esophagus में क्या रहेगा? Adventitia रहेगा. Why? Why? Common sense, because most of the part of the esophagus is present in thorax. Some part is present in the neck. वहाँ पे peritoneum नहीं रहता है. Common sense. That's why mostly it will be made up of adventitia. कौन सा part में serosa होगा? Which part? The abdominal part. Okay, approximately 2 centimeter, 1.5 to 2 centimeter जो length है, where it is attaching with the cardiac end of the stomach. That particular part only will be covered by peritoneum, and in that part you will be getting the serosa. Why I am telling you this? Because serosa mostly it is not seen. Okay, mostly you will get the adventitia. Okay, कैसे differentiate होगा serosa adventitia में? No, microscopically. सीरोजा में क्या रहता है? जस्ट नाउ आई हैव मेंशन मेसोथेलियम रहता है, सिंपल स्क्वामस एपिथेलियम, जो एडवेंटिशिया में नहीं रहेगा. ओके, सो दिस आर द लेयर्स. नाउ आई होप यू कैन सी द डायग्राम. Everyone, can you see the diagram? Yes. Now in the diagram, can you tell me from which part, which is the innermost layer and which is the outermost layer? And please remember the layers which I have mentioned. जैसे ये mucosa, this is the innermost layer, and adventitia or serosa, this is the outermost layer. Now here, can you tell me which is the innermost and which is the outermost layer? Okay, that means this is the this is the innermost layer, and this is the outermost layer. ठीक है? So like this you have to go. That means whenever we will see, whenever you will focus the slide, first which layer you have to see? You have to see the mucosa, the innermost layer. Gradually you will come to the outermost layer. Okay, same thing you have to follow whenever you are writing in the exam. First you have to mention about the mucosa. Gradually you have to go outside. Okay. Now in mucosa, how many layers are there? 3 layers are there. Lining epithelium, lamina propria and muscularis mucosae. ठीक है? Now in lining epithelium of the esophagus, what is the lining epithelium? As I said, there is a exception. Stratified squamous non-keratinized epithelium. How you will differentiate between the stratified squamous non-keratinized and simple columnar epithelium? कैसे differentiate करोगे? How you will differentiate? Stratified का मतलब क्या है? Multiple layer. Multiple layer means you will see that multiple nuclei are present. As simple as that. Can you see multiple nuclei? If it is simple columnar epithelium, तब क्या दिखाई देगा? Nucleus that also in one layer, that also at the basal part of the cell. Okay, like this you have to identify, you have to do the differentiation. Why I am telling? Because there is a point that is called cardio-esophageal junction. That is the junction between the esophagus and the stomach, cardiac end of the stomach. If you see the microscopic structure of that particular area, there you will see both the epithelium. That means you will see the stratified squamous non-keratinized epithelium. Along with that, you will also see the simple columnar epithelium. At that time, you have to differentiate that yes, this is the stratified squamous and this is the simple columnar epithelium. Okay, so you have to understand the difference. Then only you can differentiate. Okay, next, I hope you can see the lamina propria. ये देखो lamina propria is there. Now, next to the lamina propria, can you see a smooth muscle layer is present? Can you see the smooth muscle layer? This one, ये वाला, yes, this one. This is the muscularis mucosae. Okay. Now, in order to see the muscularis mucosae of esophagus, you have to take the cut section of the lower end of the esophagus. Why? Because in upper end and the middle of the esophagus, skeletal muscles are there. So appearance will be different. Okay, so that's why in order to see the muscularis mucosa, what you have to do, you have to take the cut section of the esophagus of the lower end or the terminal end of the esophagus. Then only you can see the muscularis mucosa, because in that particular area only smooth muscle cells are present. Okay, so this is the muscularis mucosa. Okay, next, which layer is this? Which layer is this? Submucosal layer. Okay. In case of esophagus, जो submucosa होता है, in that we are getting the glands, esophageal glands. Here you can see esophageal glands. So this kind of appearance will be there in the microscope that you have to identify. Okay. So these are the cut section of the esophageal glands, mucus secreting glands. Next, outside of that you can see this is the muscularis externa. तो muscularis externa में mainly in case of esophagus दो layer ही रहता है, that is the inner circular and outer longitudinal. So you can see this is the inner circular, this is the outer longitudinal. Okay, beyond that you will be getting the adventitia, which is mainly made up of connective tissue where large nerve fibers, blood vessels, lymphatics are present. Okay. So lining epithelium, it is stratified squamous non-keratinized. Okay. Next, lamina propria consists of loose connective tissue with papilla. This is very important. Okay, this is a unique feature in case of esophagus. Okay, तो जो lamina propria होता है, the lamina propria of the is mainly made up of loose connective tissue, but it is having papilla. Now papilla is nothing but finger-like processes. Okay. Now this particular papilla which is present, finger-like processes of the connective tissue, project into the epithelial layer. Okay, so they will go and they will project into the epithelial layer. This helps to prevent the separation of the epithelial from the underlying connective tissue. Okay, so the lamina propria ऐसे epithelial में अंदर घुसेगा. Okay, finger-like projection, they will go and they will get inside the lining epithelium. Okay, and they will hold the lining epithelium so that it cannot be separated. Because continuously whenever we are swallowing something, drinking something, friction is taking place. Okay, due to that friction there may be separation of the layers. That's why this particular papilla is given. Okay, this particular papilla is given, which helps in holding the epithelium, prevents the separation of the epithelium from the connective tissue. Okay, so this is very important whenever you are writing about the esophagus in practical notebook, don't forget to mention this. Next, muscularis mucosa, only longitudinal smooth muscle fibers is there and it is thicker compared to the other parts of the gut. Next, submucosa, dense irregular connective tissue as usual, along with that bundles of collagen fiber, elastic fiber, all those things are present. Okay. Now in case of esophagus, because of the presence of the fibers, these elastic fibers, collagen fibers, star-like appearance can be seen. Now what is the meaning of this? Whenever you will see the lumen of the esophagus under microscope, इसका जो appearance होगा वो star-like appearance होगा. Star-like appearance can be seen. That is because of this particular fibers which are present in the submucosa. Okay. Next, it also contains mucus secreting esophageal glands, which just now I have shown you. Next, muscularis externa, composed of striated muscle in the upper third, mixed in the middle, and smooth muscle in the lower third of the esophagus. That is the exception in case of esophagus. Adventitia, connective tissue is there with capillaries, and lower 1 inch or approximately 2.5 centimeter of the esophagus is covered by peritoneum, where you can see the serosa. Remaining entire part is covered by adventitia. Okay, so these are the layers which are present in the esophagus. Okay, so just see the layers of the esophagus. Mucosa, stratified squamous, non-keratinized. Lamina propria is there. Okay, muscularis mucosa, only longitudinal smooth muscles are present in case of esophagus, which is very thick. Next, submucosa, dense, irregular connective tissue, elastic fibers, then collagen fibers are also there, and because of those fibers which are present, star-like appearance can be seen. Next, yes, you can see star-like appearance, and then the esophageal glands which are present, okay, mucus secreting glands which are present, it is compound tubulo-alveolar glands. It is a classification of the gland. Okay. Next, muscularis externa is there. So upper third skeletal, middle mixed, and lower part only smooth muscles are there. And adventitia, mostly it is made up of connective tissue. The lower 1 inch or approximately 3 centimeter is covered by peritoneum, where serosa is present. Okay. This is the schematic diagram which you have to draw in your practical notebook. I will request everyone to please note it down. Every time whenever you are submitting your practical notebook, I don't know from where you are drawing the diagrams. Okay? So please note it down, and this time I don't want any mistake while drawing the practical diagrams in histology notebook. Now what you can see over here, anyone can take the photo and share in the group. You don't have a common group, WhatsApp group? हाँ, तो कोई भी एक लेके फोटो डाल दो WhatsApp group में, हो गया. Now what you can see over here, so where is the mucosa? Yes, last bench, where is the mucosa? Where is the mucosa? See, mucosa is there. So, and what is this? This is the lining epithelium. After that, I hope you can see the lamina propria. ये देखो, this is the lamina propria. Then this is the muscularis mucosae. Then this is the submucosa, where you can see the submucosal glands. ये देखो, these are the submucosal glands, or also called as esophageal glands. What is the classification? Compound tubulo-alveolar gland. Compound tubulo-alveolar gland. Next, here you can see muscularis externa. Okay, see, this is the muscularis externa. Okay, and then this is the adventitia. Okay, so this is the schematic diagram for drawing. This you have to draw in your practical notebook. Next, another diagram is here. Now this is the histological diagram. This is under low magnification. This is under high magnification. ठीक है, तो हाई मैग्निफिकेशन में क्या आइडेंटिफाई करना होता है? व्हाट यू हैव टू आइडेंटिफाई अंडर हाई मैग्निफिकेशन? You have to identify the lining epithelium. See, which epithelium is this? Which epithelium is this? Stratified squamous, non-keratinized. Okay, because you can see nuclei are present in multiple layers. That means it is stratified. Okay, that can be identified easily under high magnification. Now in low magnification, all the layers you can see. Next, as I have mentioned, whenever you will see the esophagus under microscope, what will be the appearance of the lumen? Star-shaped appearance. Okay, ये देखो थोड़ा सा star-shaped appearance आ रहा है. Okay. Next, directly we'll go to the stomach. This is all for the esophagus. Layers will be there. 4 basic. Yes, last bench. How many layers will be there in the stomach? How many layers will be there in GIT? Stomach is a part of GIT, right? Then how many basic layers will be there in the stomach? 4 basic layers. So I hope you can see the diagrams. Now in histology of stomach also same 4 basic layers are there. Lining, see mucosa. Mucosa is made up of 3 layers. Lining epithelium, lamina propria, muscularis, mucosae. Okay. Now do you remember the modifications which I have mentioned in case of stomach? First, can you tell me what will be the lining epithelium? Yes, yes, yes. Simple columnar epithelium, the lining epithelium of the stomach. ठीक है. Next, I have told you that जो muscle coat है और muscularis externa है, mainly 2 layers रहता है. But in case of stomach, it will be 3 layers. Additional layer is there. That is the inner oblique, then circular, then longitudinal, which can be seen easily under microscope. Okay. Next, the lamina propria, which is there. Mostly it will be occupied by the glands, gastric glands. Okay. Now, if you see the diagram carefully, can you see the lamina propria over here? Can you see the lamina propria? ये देखो, this entire part is the mucosa. So we know that in the mucosa, which is the last layer, muscularis mucosae. So see, this is the muscularis mucosae. Okay. Now this entire part is the lamina propria, and the lamina propria is packed with gastric glands, from which the secretions are taking place. Okay. So these are some modification which you can see. Now can you see the muscular layer, the muscularis externa? ये देखो, 3 layers are there. Oblique layer, circular layer, and longitudinal layer. Okay, 3 layers will be there. And we know that stomach is a intraperitoneal organ. Stomach is an intraperitoneal organ. That's why we will be getting serosa. I am not telling that entire stomach is covered by peritoneum. You know some bare areas of the stomach. Okay, so mostly the section is not going through that particular part. So from wherever the section is going, serosa will be present. So in case of stomach, you don't have to mention about the adventitia, mention about serosa. Okay, so the lining epithelium is columnar and mucus secreting. Okay, the lining epithelium of the stomach is simple columnar epithelium, that also mucus secreting cells. The apical part of the cells is filled by mucin, and usually it is removed during the processing. That's why it looks empty. What is the meaning? We know that columnar cells are taller cell. If you compare the columnar cell and cuboidal cell, which are taller? Columnar cells are taller, and the nucleus are present at the base of the cell. Okay, तो जो ऊपर वाला जो part है cell का, especially in case of stomach, what is present? Mucins are present. Whenever the preparation of the slide is done, they are washed away, they are washed out. That's why that particular part of the cell will appear empty. Additionally, जो goblet cells वगैरह नहीं रहता है, okay, from that part only the secretion is taking place. Okay, but under microscope it will look empty. Why? Because during the processing that mucin part is washed away. Okay, in order to see that particular part, a special type of stain is there, and that is called as PAS, periodic acid skiff stain method. Okay, periodic acid skiff staining method. It is a method of staining. If you stain with that PAS method, then you can see the mucin which is present at the surface of the cell, means inside the cell. Now mucus secreted by the cells of the lining epithelium protects the gastric mucosa against acid and enzyme. As simple as that. Already we know that. Numerous places the lining epithelium will dip into the lamina propria. Now there are various areas where the lining epithelium will go deep into the lamina propria, and these are called as gastric pits. So whenever it is going downwards, तो क्या होगा? A depression will be produced, and these are called as gastric pits. You have not studied the interior of the stomach, huh? Then in interior of the stomach you have not studied gastric rugae. Yes. In between the rugae you have not studied the pits. So I am talking about those pits. Now these pits extend from variable distance into the thickness of the mucosa. Okay, that means the thickness of the pits are not same. Some are having more thickness, some are having less thickness. Okay, now deep to the gastric pits, the mucous membrane are packed with numerous gastric glands. This is very important. That means stomach का जितना secretions होता है, those are coming out from these gastric glands. Okay, so deep to the pits, it is filled by gastric glands, and from there only the secretions are taking place. How many types of glands are there in the stomach? 3 types of glands are there. What are they? Main gastric gland, cardiac gland, and pyloric gland. Okay, majority of them are the main gastric gland. Majority of the stomach में जो glands रहता है, these are called as main gastric gland. In cardiac part we are getting the cardiac gland, and pyloric part we are getting the pyloric glands. Okay. Mostly they are mucus secreting glands, and rest other secretion whatever it is required, these are taking place from the main gastric gland. Okay. Now see, I have noticed that in most of the books, okay, in order to study the stomach, 3 separate parts are given. Okay. For example, histology of cardiac part, okay, then fundus or body, and the pyloric part. In most of the books, okay, if you I don't know which book you are referring, in most of the books how it is given, histology of first cardiac part, then fundus or body, and then pyloric part. Like this it is given separately. Okay. Now if you study everything, okay, and later on if you draw the conclusion, okay, then whatever I have noticed is the difference is mainly in the glands. Okay, the difference is mainly in the glands, and the differences are in the muscularis externa. Remaining everything same रहता है. Okay, main difference कहाँ पे रहता है? Glands. That also in the secretion, जो secretion होता है. Okay, then in the type of cells which are present. Okay, so overall the differences are in the glands, and second difference is in the muscularis externa. These are main 2 differences whatever I have noticed. Okay, so that's why what we'll do, we'll discuss the histology of stomach overall by taking all the parts in hand. We'll be discussing, and after that we'll draw a conclusion in a form of chart, so that we can draw the we can make the differences कि pyloric end में क्या रहता है, gastric end में क्या, body में क्या रहता है, and cardiac end में क्या रहता है. Okay. Now the majority of the glands are the gastric glands. Okay. Now these gastric glands which are present, most of the stomach, the gastric glands are present. Okay. Now the main gastric glands are simple or branched tubular glands. The classification, if you see, simple or branched tubular glands, and they are lying right angles to the mucosal surface. Now the gland open into the gastric pits, each pit receiving opening of the several glands. तो जो gastric pit होता है, below the pit glands are there. Now that particular pit can receive secretion from multiple glands. Okay, तो जो gland होता है, pit होता है, दोनों के बीच में जो area होता है, that is called as isthmus. Okay. Pits are there, that means the surface depression. इसके नीचे क्या रहता है? Glands रहता है. Okay, secretory cells. So the difference, the area which is there, that is called as isthmus in between this. Now what are the important cells present? First, you know that chief or zymosome cells are there. Already you have studied in physiology, I know. So these are cuboidal cells or low columnar cells. Basophilic cytoplasm are there. Okay, they are located in the deeper or basal part or lower 1/3 of the gastric glands. That means the location of the chief cells or the zymosome cells, where they are present at the basal part or lower 1/3 of the gastric glands. Okay. Now what is the function? Chief cells secrete digestive enzymes of the stomach, including pepsin. What is the function of the pepsin? Digestion of protein. That means it will convert the protein, or it will break down the protein into peptides. Okay. After that the digestion can take place easily. So you can see pepsin break down the protein into small peptides which are required for digestion. Next, the apical portion of the cells having secretory zymosome granules, okay, slight apical eosinophilia. If you see the electron microscopy of this particular cells, okay, then abundant ER is there, basal part, and secretory granules are present in the apical part, and luminal surface shows numerous small microvilli. Okay, so you can see luminal surface shows microvilli, and then here you can see the granules which are there, zymosome granules. From here only the secretions are taking place. Okay, so this is the schematic diagram of electron microscope image of the chief cells. Next, here you can see the parietal cells also, and you can see the enteroendocrine cells also. Next, parietal cells are also called as oxygenetic cell. Okay, so what is the shape and size? It is ovoid or polyhedral in size. Large central nucleus is present. Now these are slightly sandwiched between the peptic and the basement membrane. Okay. Now where these cells are present exactly, more numerous in the upper half of the gland. Previously we have seen those chief cells where they are present at the lower part of the gland. But these particular cells, they are present at the upper half of the gland. Now they are called as oxygenetic cell because they stain strongly to the eosin. And what is the function? What is the function of these oxygenetic or parietal cells? They secrete HCl. Okay, now if you see the electron microscope structure of the parietal cell, the apical part reaches the lumen of the gland. Okay, the cell membrane shows several invagination into the cytoplasm, producing tortuous intracellular canaliculi. Through that canaliculi only the secretions are coming out. Now the walls of the canaliculi bear microvilli which project into the canaliculi. Okay. Now these oxygenetic cells, they secrete HCl. Along with that, they also secrete the intrinsic factor of Kessel. Okay, intrinsic factor of Kessel. Now this is very important because it helps in the absorption of vitamin B12. Okay, or else there will be a condition that is called as pernicious anemia. Now here you can see the EM structure of the oxygenetic cell or parietal cell. Okay, so these are the intracellular cleft with microvilli. Okay, so the secretions are coming out through this intracellular cleft. Okay, next this is a 3D diagram where see these are the gastric pits. Now deep to the gastric pits you can see the gastric glands are there. Okay, so gastric glands से जो secretion होता है, it used to come out like this. So this particular part is called as isthmus. Okay, next other layers also you can see, all the layers whatever I have mentioned. Next other types of cells are also present, that is the mucous neck cell which are present at the neck of the gland. Then these are mucus secreting cell. Okay, now please note that two different types of cells are there which are secreting mucus. One is the lining epithelium and another is the mucous neck cell. Now what do you think, what will be the difference between this? So we have seen that the lining epithelium is also secreting mucus, and mucous neck cells are also there which is present at the neck of the gland. They are also secreting mucus. What do you think, what will be the difference between these two mucus? Concentration, not exactly. Any idea? And what about this one? No. Please find out what is the difference. I am giving you a hint. Okay, one mucus which is secreted by this one. Okay, this lining epithelium, the mucus which is secreted, it is insoluble, and this is soluble. Now try to find out what is the difference. Differences are there, that's why I have asked you. ओके डिफरेंसेस है इसलिए, नहीं तो एक ही सेल रहता है, दो अलग अलग सेल है, दोनों से म्यूकस सीक्रेट होता है, उसका जो कॉन्स्टिट्यूएंट है वो अलग होता है, along with that जो फंक्शन है that is also different. Okay, please find out this one. Okay, next enteroendocrine cells are also there. Now these enteroendocrine cells are containing membrane bound neurosecretory granules, very important. Okay, they are also called as argentaffin cell. Why they are called as argentaffin cell? Because they can be stained with the help of silver salt, that's why they are called as argentaffin cell. Now what is their function? As you can see, they secretin, gastrin and histamine. Okay, secretion of gastrin, serotonin and histamine. These are the hormones which are secreted. They are having very important function. For example, the serotonin which is there, it is a neurotransmitter. Okay, it is a neurotransmitter for enteric nervous system. Then this gastrin which is there and histamine, okay, दोनों का opposite function है. जो gastrin है, what it will do? It will inhibit the secretion. Okay, और जो histamine है, that will enhance the secretion. Okay, now I hope you have heard about a drug that is called as ranitidine. Anyone, have you heard about that drug called ranitidine? Okay, one of the largest selling drug. Okay, so this particular cells, they are having a receptor that is called H2 receptors. ये जो particular cells है, these cells are having a receptor on their surface. These are called as H2 receptor. Okay, now whenever we are taking this ranitidine, what they will go and bind to the receptor, and after that they will inhibit the secretion. Okay, तो histamine secretion अगर inhibit होगा, then what will happen? The overall secretion will get reduced. So this is the way how the drugs are acting. Okay. Next, some undifferentiated stem cells are there. So already you know what are the functions of the stem cells. Okay, if any damage is there to the other cell, they can replace them, and they play an important role in healing. Okay, now this is the overall gist, whatever we have discussed about the cells present in the gastric gland. Okay, so you can see the mucous neck cell, chief cells or zymosome cells, parietal oxygenetic cell, enteroendocrine cells, and the stem cells. Okay, so everything is given, location, identification, identifying feature, that means what is the shape of the cell, then what are the functions, lifespan means how many days it can survive, and then about each and every cells, all these points are given. Okay, so I'll request everyone to write this particular part also in the practical notebook. Okay, next, if you see the cardiac glands and the pyloric glands, as I said, the differences are very less. Okay, mainly they are mucus secreting glands. Now these particular cardiac glands, where they are present around the opening of the esophagus or around the cardiac end. Now here also the gastric pits are there, but the gastric pits are shallow. Okay, so everywhere, if you see the cardiac end, if you see the pyloric end or the pyloric part, gastric pits will be there, but there are difference between the gastric pits. Okay, तो जो cardiac end है, in that particular part gastric pits are very shallow. That means जो depression होता है, that is shallow, it is not deep. But if you go to the pyloric one, the depression is more, means it is more deep as compared to the cardiac one. So from there also it can be differentiated. Next, cardiac glands are either simple tubular or compound tubulo-alveolar. Okay, and these are mucus secreting. Occasionally oxygenetic cells or peptic cells are present, but mostly they are absent, mainly mucus secreting. Next, in case of pyloric, it is branched tubular glands or coiled. That is the difference. Now glands are lined by, again see, mucus secreting cells. Pyloric glands also mainly mucus secreting cells are present. Okay, now like this we can do the differentiation. Another differentiation is the pits which are there, it is more deep in case of pyloric gland, and in case of cardiac gland it is very less. Now in between them, if you see the gastric glands, it is in between the cardiac and the pyloric gland, the depth of the pits. That means जो mucosal lining है, mucosal lining से it will go downward towards the lamina propria. Okay, so उसका जो depth होता है that is more in case of pyloric one. Average होता है gastric gland में और सबसे कम होता है cardiac glands में. Okay. Now why I am telling you this? Because this can be seen under microscope. That's why. Okay. Next, in the beginning, I hope you remember that I have shown you one diagram of the glands. Now, if you see the lamina propria, so whatever we have discussed earlier in the layers of GIT, same thing is present in the lamina propria, but the amount of connective tissue is very less in the lamina propria of the stomach. Why it is less? Because it is packed with the gastric glands. Most of the areas are packed with gastric gland. That's why you can't see the connective tissue. It is there, but it is scanty, very less. Okay, that is the lamina propria. Next, muscularis mucosa, which is there. Okay, muscularis mucosa. Now that particular layer is well developed in case of stomach. Why it is well developed in stomach? Because stomach is also having its secretion. From stomach continuously the acid is secreting. Okay, and along with that other enzymes are secreting. Okay, so whenever we are eating something, it goes to the stomach. It has to mix with that content. For that, the gastric mucosa which is there, the muscularis mucosa, that is well developed in stomach. Okay. Now the submucosa in case of esophagus, I hope you have seen where the glands are present, compound tubuloalveolar glands. But here in case of stomach, glands are absent. कोई भी glands नहीं रहता है in case of submucosa. Okay. Instead of that, lymphatics are there, blood vessels are there, and in submucosa, one nerve plexus is present. Which nerve plexus is present in the submucosa? Meissner's plexus, that is present in the stomach. Okay, so don't forget to mention whenever you are writing. Next, what about the muscularis externa in case of stomach? जो muscularis externa रहता है, in muscularis externa of the stomach, 3 layers of muscle fibers are there. First will be the oblique. Second is circular. Okay, so these 3 layers are present. Okay, in the stomach. That is the speciality of the stomach. जो muscularis externa है उसमें 3 layers रहेगा. Along with that, what is present in the muscularis externa? Which nerve plexus is there? Myenteric plexus is there. Okay. Next, do you remember the modification which I have mentioned in case of stomach? जो muscularis externa है, muscularis externa का जो circular layer है, okay, it is getting thickened at the pyloric end to form the pyloric sphincter. As simple as that. Okay, so that also you have to mention. And after muscularis externa, what is the next layer? Yes, serosa. So in case of stomach, don't write adventitia, it will be serosa. Serosa में मतलब what you will write in serosa? It is covered by peritoneum. Okay, so that is all about the stomach. Wait, let me show you the diagram and then we'll see the slides. Points. Okay, so here you can see cardiac part of the stomach. So in the beginning I have mentioned in most of the books, the way they have given, it is divided into 3 parts. It is first the cardiac part, then the fundus or the body of the stomach, and then the pylorus of the stomach. Okay, 4 basic layers will be there. Where the differences are mainly in the glands. Okay, and the differences are present in the muscularis externa also. Okay, so please note it down. So this is very important. This will help you to understand between the differences of various parts of the stomach. Okay, next this is the comparison. Okay, where you can see the cardiac part, fundus of the body, and the pylorus of the stomach. Now this is also important. Please note it down, this particular part. Now let's see the diagram, and after that we'll see the slides. So what do you think? Which part is this? Which part is this? Which part of the stomach is this? Cardiac part. Okay. Now here you can see cardio-esophageal junction. Can you see the esophagus, part of the esophagus? That's why you can see stratified squamous non-keratinized epithelium. And here you can see the simple columnar epithelium. Okay, this is the schematic diagram. Now, as I have mentioned, the pits which are present in case of this particular part, cardiac part, it is shallow. You can see the pits are very shallow. It is not deep. The pits are shallow. Okay, this is the schematic diagram, and this is the original histology diagram. Next, here you can see the fundus of the body, where you can see pits are there. It is not that shallow, not that deep, average. Okay, means this you can easily differentiate under microscope. Okay, where you can see all the different types of glands. In the gastric glands, you can see the parietal cells. You can. Next, this is the histology of the pylorus of the stomach. Can you see the depth? Okay, see the depth of the gastric pits. It is more as compared to the other part. So from there easily you can differentiate. Okay, because देखो ये जो particular cells है, okay, those cells which are present in the gland under compound microscope very difficult to identify. For that special stains are required. ठीक है, but these pits can be seen. ये देखो pits can be seen easily. From there you can identify. Okay.